All right. Thank you, everybody, for joining us this afternoon. We appreciate you taking time as we listen to Dr. Troy Ott with Penn State College of Agricultural Sciences give us an in-depth look at animal biotechnologies, sort of what has happened, what is happening, and um, what is yet to come as far as future technologies to advance animal agriculture. So, Dr. Ott, it is all yours.
Good afternoon, and that is certainly a large task that you've put up there for me, John. I wonder if you will help me keep an eye on the questions, so when I share my screen, I might not see a raised hand or something in the chat box. So if you see anything, if you could just break in. Also, I'm perfectly comfortable with anyone just unmuting themselves and interrupting me. The favorite question I like to answer is the one that you have on your mind at the moment. And so I'll talk about some things today, but I may miss some things that are important to you. And I'm perfectly happy to go off down any tangent you'd like to. So don't hesitate to interrupt and ask me a question. So I'm going to go ahead and share my screen. Let's see how this works. And then I'm going to put it on like this. Okay. So Neil, can you see my screen? Okay, good. Excellent.
Yeah.
So we got a big topic here, biotechnology and animal agriculture, and so I'm going to move very quickly because I'm going to move very quickly through some things before we actually talk about biotechnologies. I think it's always important to talk about why it is that we are wanting to apply technology to animal agriculture. Certainly as a scientist and a teacher, it's easy to get excited about technologies, but I think one of the most common questions that I get when I talk to various publics about these are, why are we using technology? Why do we have to use technology? Why are we messing with agriculture, which seemed to be perfectly fine the way it was, or the way I remembered it to be? And so I think agriculture has this endless struggle with the way it used to be, and people's perception of the farm in the good old days versus today. You'll hear the term industrial agriculture. It's used as a pejorative, but I can tell you, I had the benefit of being raised by my grandparents, and my grandfather's family dairied in the Philadelphia area about the turn of the 20th century, so late 1800s, early 1900s. And I used to talk to Pop a lot about farming back in those days. And that picture you see on your left is would have been probably seen on the farm that he grew up on and worked on. He used to tell me, he said, people who want to go back and farm the way we did back then don't know anything about what farming was like back then. It was tough, and it was a lot of hard work, morning to night, 7 days a week often. And so he was he had a fairly modern vision for technology. So I think because of the fact that many of us, most of us are a couple of generations removed from the farm, we are stuck with this idea of the way it was and how did it get screwed up along the way. I don't know if you can think about the last time you heard someone talking about agriculture, but almost always we hear this story about this broken food system that we have. And I've never seen the broken food system they're talking about. I've seen one of the most efficient systems of producing and delivering food that's healthy and low cost that we've ever had in the history of humanity. But and talking to certain people, you think that we were at the front edge of a train wreck. So when we talk about technology, I think when you talk about technology, you need to frame the issue carefully with your students and your audience. Why is it that technology needs to be applied to animal agriculture? And I do the same thing when I give talks to the general public. And so I always, because I'm a reproductive biologist and I study fertility and infertility, I like to start with the obvious challenge that's on this slide here, and that is that population is growing, and it's going to continue to grow for some period of time. And I wanted just to give you a sense for how much population has grown. You see this little red arrow here on the bottom. This is the year I was born. This was the year I was born in 1962. And just in my lifespan, the population of the planet has more than doubled in my lifetime. And we're going to add about the equivalent of 2 more Chinas between now and 2050. 2 more Chinas between now and 2050. And most of this population is going to appear in places that already struggle with hunger, that have harsh climates and poor soils. And so we have a challenge that is not an abstract challenge. It's a real challenge. And we need to be training the next generation of agriculturalists, farmers, and ranchers to be able to meet that challenge. Now, it's not enough of a challenge that we need to produce 50 to 70% more food with roughly the same amount of land and less water, but we now have to do this while reducing the impact of farming on the environment and improving the welfare of the animals that we raise. So this is a real challenge, and it will require really serious people working very hard to meet this challenge and to meet the expectations of society. So when you break it down in terms of animal agriculture, you can see that the predicted demand and increase in global consumption of animal protein up to 2030, primarily pork, chicken, and fish, but also beef. And you can see that demand for animal protein is increasing in other countries. In particular, it's not just the population growth that's driving this increase in demand. As people get wealthier, they spend more of their disposable income on animal protein. And so one of the untold stories of the last 30 years is the tremendous reduction in poverty, and people have gotten their people's wealth has increased. And as they have more money, more disposable income, they're going to consume more animal protein because it's a high quality source of protein in their diet. So this is a real challenge. Like I said, society also continues to raise the bar on agriculture in terms of how we farm the land and how we raise the animals. And in particular, right now, there's a tremendous focus on greenhouse gas emissions and climate change. And it's important that agriculture accepts its responsibility. But I think it's also important that as you have a discussion about greenhouse gas emissions, that we frame it correctly. And if you look at greenhouse gas emissions in the United States, you can see here that agriculture accounts for roughly 10% of greenhouse gas emissions in the United States. And these are data from 2018 from the EPA. Now, that's certainly something that we have to take ownership of. And if you break that down, about half of that is from animal agriculture. And so we have a piece of the pie that we have to address. And we're going to have to do that. We're going to have to reduce the environmental impact. We're going to have to reduce greenhouse gas emissions. And we're going to have to do that while raising the welfare of the animals that we grow. So this is really an important and difficult challenge. So the question is, can we get from here to there? And are we going to be able to not only produce more food, but also address the environment and animal welfare conditions? This is the frame that I think that we can use to discuss technology. As we talk to various audiences, to students, everybody wants safe, affordable food. Everybody wants reduced environmental impact. And so everyone wants improved animal welfare. So what do we have to do to get from here to there? Well, technology has a number of solutions for some of the problems that we're facing. And I'm going to talk a little bit about how some technologies have been used. And I think I know I'm preaching to the choir because many of you are familiar with many of these technologies. But if you understand human nature, you understand that people tend to fear things that they don't understand. And this is a cartoon taken from around 1900, depicting this particular artist's concern about how electrifying our cities was going to result in a spider web of death. And this was truly reflects some of the impressions that people had. And if you go back, and many of you may have done this, everything from the invention of the printing press to pasteurization of milk to vaccines, every new technology has been greatly feared in the beginning. So this is what happens whenever we apply technology in any aspect of human activities. There's an immediate fear, and then over time, people can become more comfortable with those technologies. So regardless of the technology, there's always a hype cycle and a life cycle. And, I mentioned electricity. I mentioned pasteurization of milk, microwave ovens, cell phones, you name it. There is this hype cycle and life cycle, and we're seeing it in agriculture even today. And the way this works is, we have innovators in society that are thinking about how we can do things better. Many times, those innovators are thinking about how they can become wealthier by solving a problem. And innovation is built into the fabric of this country. Article 1, Section 8 of the U.S. Constitution outlines patent law, because everyone, even back a couple hundred years ago, understood that inventors needed to be able to benefit or to reap the fruits of their invention if we were going to be innovative and if we were going to solve the challenges that faced humanity. So innovators come up with ideas. Typically those innovations oftentimes come as a result of some advance in technology. And then there's this curve of expectations, and we call it the hype curve, a hype cycle. You see this very often. I'll give you a classic example of where the hype cycle is run amok right now, and that's in the area of the technology of cellular agriculture or in vitro meat. That many people, many companies, investors are investing hundreds of millions of dollars in cellular meat right now, and there will be a market at some point in the future. But the expectation is really running rampant, and a lot of people are investing a lot of money in growing cells in culture, and then the idea that they would form them into various meat products that people would buy. So this is what we have to deal with. It is the expectations, and usually after those expectations, after we enter through that hype cycle, there's a trough of disillusionment. And then if the technology actually meets a need and fits some other criteria, slowly over time it becomes adopted. And in general, throughout our modern history, we can see most new technologies take about a generation in order for them to be widely adopted. And if you look historically at some of the technologies that you'll be familiar with, things like the telephone, electricity, in general, many of those things took many decades before they reached, say, 80% adoption in society. Others moved more quickly, and they moved quickly because of the need they filled. For example, if you look at the automobile here in around 1915, you can see how we went from almost no automobiles to roughly 60% adoption in a really short period of time. Anyone know why it flattened off and declined there for a little while after in this peak here in around 1929, 1930? Anyone can guess why it didn't continue to rise?
The Great Depression?
The Great Depression. Was that Amy? Yeah.
Yes.
Good. So Amy gets a star, John, if you're giving stars out. So yeah, so look at other things that really fit a need: refrigerations, clothes washers, other things that move very quickly are color TVs. And as we move to the present, you can see many of these newer technologies have a much more rapid adoption cycle. But in general, many of these things take about a generation for people to become comfortable with the technology. And I'm going to talk a little bit about gene editing in my talk today. And it's in agriculture, it's been a very rapidly adopted technology in plant agriculture. And so it is probably one of the most rapidly adopted technologies that we've seen in a while. So when we start to think about applying technology, and framing, why it is we should do this, we need to think about what is the need. Can we provide access to this technology, and is it affordable? And that's going to drive this adoption cycle. So I mentioned for any new technologies, there's always controversy and there's always some fear associated with it. And this is, as Hugh Grant pointed out, this is just the price of being first sometimes. One of the problems with technology is that humans tend to imagine the worst case scenario of any technology. So when something new is presented, we in our mind think about what is the worst case scenario that could happen. And in general, the people that study this type of human behavior understand that if we can imagine it, we assign things a very high probability. All we have to do is be able to imagine something, and we assign it in our minds a very likely probability. In fact, we will ignore actual data on the probability of that bad thing happening, because as long as we can imagine it, we think it's likely to occur. And I think if we look at the European Union and how they've applied the precautionary principle to adoption of agricultural biotechnology, particularly genetic engineering, that's a very extreme example of this type of risk assessment, where you have to prove something is safe before it can be adopted. And as you know, science doesn't do that. What science can do is can assess what the risk is and put that risk in context with other risks in that same category. So, you know, when someone says, can you prove to me, or can you guarantee something is never going to go wrong? Scientists can never say that. That's not how science is done. So let's talk a little bit about some technologies in animal agriculture, and maybe I'll pause here and take just a deep breath. If there's anything that I said up to this point that stimulated a question or a comment, please unmute yourself and jump in. All right. So let's talk about some technologies in animal agriculture. And John knows I'm a reproductive biologist, so I'm going to talk first about one that's been adopted here in the recent past, within the last 15 years, that has had a big impact on at least certain animal agriculture, and that is the technology of sexed semen. And sexed semen has really come to the forefront in about the last 10 years. And I'm going to go through the procedure and how it's done. If you have any questions as I go along, please interrupt. But this has really been adopted at a fairly high level in the dairy industry. As you might imagine, if you're trying to grow your dairy and expand your dairy, which many dairies are, you need to have daughters. And so by being able to purchase semen that has been gender selected, you can do that. Likewise, now as people are thinking about putting beef semen into dairy cattle, into their lower ends, at the lower end of their genetic herd, in terms of the lower end of their cows, you could put in only male semen in those, so you get steers that would be more valuable to fatten. So this technology has a number of applications. One of the things, though, it did is it really flooded the market with heifers, and it really created a problem with heifer prices. And so the market had to adjust to that disruption. But it has brought a lot of positive change. So basically, sexing sperm is based on the simple principle that if you compare the Y chromosome and the X chromosome, you can see the Y chromosome is very small compared to the X chromosome. And so a sperm will only contain either an X or a Y because it's haploid. And so a sperm that contains a Y chromosome has about 3.8% less DNA in it. 3.8% less DNA in a sperm that has a Y chromosome versus one that has an X chromosome. Now, that doesn't seem like a lot, but we have some fairly sophisticated technologies that allow us to differentiate between that. And the principle is quite simple. We just take the sperm that's ejaculated, we dilute it, and then we stain it with a dye that binds to DNA. And then so we've now loaded the sperm with their DNA with a fluorescent dye, and we run those sperm past a laser here, and the laser activates that dye, and it gives off fluorescence, which we can measure over here. And the amount of fluorescence is related to the amount of DNA. So an X-bearing sperm is going to have a bit more fluorescence, shown here by more lines, than a Y-bearing sperm, which is going to have about 3.8% less DNA. So this is the basis for this technology. Well, now how do we actually, once we dye those sperm, how do we actually get them separated? And if you look at this figure here, taken from the Sexting Technologies website, basically that ejaculate in the bull might contain a billion or so sperm. Those sperm have to be diluted and dyed, as I just mentioned, and then they have to run single file through this flow cytometer. And that's a pretty remarkable phenomenon when you think about running a couple of 100 million sperm single file and actually interrogating each sperm with a laser. And the way this principle works, as I said, is it runs past the laser, and the laser will then here have a detector, and it will detect the signal. And if it's an X-bearing sperm, it will have a charging rod that will charge the droplet that it's going to end up in. And so you can see each sperm leaves in an individual droplet at the end of this stream, and that droplet will be charged. And so if it has an X charge, an X sperm in it, in this case, it will have a negative charge, and it'll be pulled towards the positive pole with a magnet here. And if it's a Y-bearing sperm, it'll be given a positive charge, and it'll be pulled towards the negative pole. If the machine can't figure it out, it'll drop down the middle and it'll go into waste. So this is the principle for sorting X and Y-bearing sperm. And the machines that do this can process hundreds of millions. That machine I showed you just a few slides ago processes 500 million sperm per hour. And they have a number of operations where they'll have 20, 30 or more of these flow cytometers running basically 365 days a year processing sperm. Just to show you what this printout looks like when it goes through the flow cytometer, you get two populations of sperm here, one that's got a little bit more DNA and one that's got a little bit less DNA. And you can see them represented here as X and Y. And you can see there's separation, but it's not perfect. And so those ones that overlap in the middle are lost. And this is the basis for sexing sperm. Now, when this technology initially came out, it wasn't perfect. And in fact, the types of conception rate you got with sex sperm was a little less than 70% of what you got with unsexed conventional semen. And this is both looking at Holstein and Jersey. You can see that the fertility, the relative conception rate was a little bit lower. But over the last 8 to 10 years, you can see that we're approaching parity in terms of we're approaching 1, which would be equivalent to conventional semen. So you can see early on there was a difference with conventional semen being a little bit higher conception rate than sex semen. But if you look here, most recently they've come together, and we've seen that in both in cows and in heifers, that the conception rates are near identical now. So when I first started teaching about sex semen, they would say only use it in heifers and only use it on natural heats because you're going to take about a 5 to 7 or 8% reduction in conception rates. That issue is being solved. So sex semen has really been rapidly adopted and has provided a tremendous amount of benefits, particularly to the dairy industry. Anyone have any questions about sex semen?
I do, Troy. And if anybody else does, please feel free to type them into the chat box or unmute to ask yourself. But when the separator segregates the sperm into its into its respective container, what percent is there a percentage of accuracy there, and what is that?
Yeah, in general, it depends on the company. And they generally say you should get sort of greater than 85 to 90% of the gender that you are buying. So, and that has gotten a little bit better. So it's probably in excess of 90%. So if you buy X sperm, you want daughters on your dairy, you're going to get an occasional bull calf, but you're going to get about 90% heifer calves. A couple of years ago, a big study was done that actually improved the value proposition for sex semen. And what they did is they looked at milk production in heifers that had two daughters in their first two pregnancies. And they found that in dairy cattle, if the first and second calf that cow has are both daughters, that she produces about 800 pounds more milk during her lactations than if she has two sons in her first two parities. So there's been even greater interest in using sex semen. The other advantage of having daughters is daughters tend to be smaller. And so when you use them on your heifers, you tend to have less calving difficulty. Other questions? So what if you can't do AI or you don't want to do AI? Are there any technologies out there for you? You may know that the dairy industry, about 80% of the dairy cows are bred with artificial insemination, but that's about 7 or 8% on the beef industry. And can anyone tell me why that is? Why is the beef industry such a low adoption rate for artificial insemination? Anyone have any guesses? No? Well, it's because...
Males and females are both producing meat.
Say that again?
I said both males and females are producing meat, so it doesn't matter as much if you're having male or female offspring.
Well, that's, but the real advantage, and you were right to think about male and female offspring when it comes to dairy cow. We want daughters and beef cows both produce a product. But really what AI allows you to do is amplify great genetics. And the reason that milk production in our dairy cows has quadrupled over the last 70 years is because we've been able to rapidly improve genetics through the being able to purchase the best bull in the country. But the problem with the beef industry is it's not very well integrated, fairly extensive, and a lot of these operations run cattle out on pasture. They don't have a lot of facilities. They don't handle these animals, so they're not really set up to do AI very well. You have to have good facilities. You have to be able to gather up your animals. You have to be able to do AI. So that's why we only really see AI on the top end of a lot of our genetics, but not on the sort of commercial cows. But I'm going to tell you about a technology that's really right on the very forefront. It's not ready for prime time, but basically, what if we could take this fantastic beef bull here, and we could amplify his genetics by putting his genetics in those those average bulls there below. This has been something that's being pursued by a number of labs over the last 20 years, and this particular gentleman here, John Oatley, who was on our faculty here at Penn State for a couple of years. John, I've known John since he was a graduate student when he took repro from me when I was out at the University of Idaho, and worked with him over the years. And his passion is to understand how you could take superior genetics and transfer those genetics into sort of grade males, and then you would basically be able to run multiple bulls that would actually be producing sperm that derived from one superior bull. So of course you can think about the challenges, and he just published a paper a couple of weeks ago which demonstrated the proof of principle in both pigs and goats. And I'll just take you through this concept, but the whole idea is if you're not going to use AI, what if we could easily transfer the sperm producing cells from a superior bull into some grade bulls, and then you could let those grade bulls run with your herd, and that way you could get the advantage of that superior genetics without adopting AI. Well, of course, you can imagine what the first challenge is. What's the first challenge if you want to do that? Anybody? What's the big challenge if you want to put new sperm into a bull? What do you have to do first? Well, you have to get him to stop making the sperm he's making already, right? You can't have a mix. We don't want a bag of Skittles. What we have to have is only the sperm from the new bull and not any of the average bull sperm. So what John did, and don't be alarmed by this slide, but I just wanted to show you the proof of principle here. What he did was he identified a particular gene, this gene here called nanos2. And if you knock that gene out, the bulls, the males are born with perfectly fine testes, except they have no sperm cells in them. So it's like a perfect apartment with no renter. And so then what you can do then is you can then isolate the stem cells from a high genetic value animal, and you can inject them into the testis, and they actually move in and they proliferate and take up residence. And so you can see what you can do here. If you look right here, you can see that they've taken some testicular tissue from a high quality donor. They've digested it down into individual cells. These cells are the progenitor for sperm. And then they've taken another male that had nanos 2 knocked out and has no sperm in his testis, and they used a fine needle and they injected these cells. And those cells will actually move into the tissue and they'll take up residence. And believe it or not, this pig produced sperm from the donor, and these goats produce sperm from the donor and only the donor. So this is a tremendous breakthrough, and you have the ability to, in fact, if you could culture these cells, you could put these cells into any number of average males that have had their regular sperm reduced or removed. So now this is really cutting edge, and so you're not going to see this for a decade or more, but it does show the proof of principle that you can actually eliminate the sperm cells from one male, replace them with the stem cells from another male, and that will then produce sperm from that donor male. Any questions about that technology? It really got a lot of press a couple of weeks ago. I don't know if you saw the news stories. I think it was even on CNN about super dads.
Yeah, I do have one question.
Yep.
Do you worry or have a fear that that is going to limit too much our genetic variation within species, that we're getting too selective if you're talking about knocking out and just going with one premium or premier sire? Or
Yeah, good question, Adam. And it would be, I think, similar to the questions that have been asked about using AI. And so if you know about AI in the dairy industry, our dairy Holstein breeds about 7% inbred. And so when you buy sperm for your cows, they do a lot of paying attention to the pedigree to reduce that inbreeding. I think this would be less of a problem. I actually, Adam, don't know that I don't know for sure whether this will ever become commercially viable, but it wouldn't be done it wouldn't be the same massive scale. Like anyone can buy the top bull in the country and really, so it is an issue that you need to think about, but I don't think it'd be any worse, and it'd probably be less of an issue than with AI. I don't know if you heard, if you trace back the genetics in the Holstein breed right now, you can trace it back to essentially two Y chromosomes. There's two Y chromosomes that represent the vast majority of the bulls that are the top 100 bulls in the Holstein breed. And so about 3 years ago, some scientists here at Penn State actually were able to get some sperm that had been frozen in the 1950s from some Holstein bulls that represented two additional Y chromosomes. So they took those sperm and they fertilized modern cow eggs, and they transferred those embryos in. And now we have some Holsteins running around that have 1950s dads and 2010 moms. And so it's like crossing a Model T Roadster and a Tesla. And so now these are interesting animals to study, to see what some of the impacts are, but also they've been able to bring these two new Y chromosomes back into the Holstein breed. So some of the technologies that some of the challenges with these technologies also provide some solutions. So we can actually now reach back and bring old genetics back in to some of our modern animal lines.
Dr. Ott, before you continue, we have a question from Ryan asking for some clarity on how that Nanos 2 gene works. Does it need to be reactivated? Because you stated it needs to be deactivated when the creation of old sperm is ceased. Does it need to be reactivated for the donor sperm to then proliferate?
No. So what they do by inactivating nanos, those testis, the germ cells. One of the things about males in any species is males produce sperm throughout their entire life, which means they have a stem cell population that continues to produce sperm. And so this, by knocking out nanos in the recipient, they don't have any germ cells in their testis. They have everything else. And when you put the donor sperm or the donor stem cells in there, they have nanos that they brought with them, and they are able to then produce sperm in the testes that they get injected into. So it's a permanent knockout of nanos in the father, in the recipient.
Dr. Ott, another question from Dr. Garby. And David, are you able to unmute and ask your question live?
Sure. Can you hear me, John?
Yep.
Great. Very cool stuff you're talking about today. I'm really enjoying listening to the information. I just had a quick question. I myself have made knockout fruit flies before, and I've seen knockout transgenic mice made. How do you make a knockout cow or pig? Is this using CRISPR technology?
Yeah, it's using gene editing and CRISPR-Cas, absolutely. And we'll talk about that in just a bit in terms of the technology. But yeah, the gene editing tools that we have now are tremendously powerful, tremendously specific, and quite easy to use, actually. Did I answer your question, Dave?
Yes, you did. Thank you.
Yep. Okay. So now we talked about sexing sperm and getting the gender you want, and we talked about how we could do that in the absence of AI with this technique. But what if your sperm are not good swimmers? Is there any technology for you there? Well, just in the last couple of months, there was a paper published titled "Toward Assisted Fertilization by Sperm Carrying Micromotors," and basically, this is a group of engineers that decided to try to develop a micromotor. And you can see an egg here in this picture, and then a sperm that has a micromotor put on it that allows it to move. Now, in cases of infertility, particularly we think about human infertility, a lot of times, about 50% of the time, it's male factor. And many times it's as a result of very low sperm numbers, or the sperm are not very motile, and so we have to move to in vitro techniques. So we have a technique called ICSI right now, where you can actually pick up a sperm with a micropipette, and you can inject it right into the egg to cause fertilization. But these guys actually went one step further, and they developed a little nanobot that can actually deliver that sperm. And so this is a little video. If it's going to work here, I'll tell you in just a second. That will, let me just see here. There we go. So it looks like a little spring that kind of would be in your pen, and they're causing it to vibrate and also moving it with a magnet. And you can see here how it's climbing, it's inserting itself on the tail of a sperm. And once it's got it on the tail, it can then drive that sperm around the Petri dish and actually drive it right up to the egg. And you can see here is an oocyte, and the sperm is being directed by this little coil. Remember that sperm is the head is about 5 microns. So this is a very tiny little motor that they've developed, and that motor can then dock that sperm right onto that oocyte without ever actually touching it with a pipette. So this type of technology is interesting and has some potential applications for assisted reproductive technologies, but also for being able to develop things that you can insert in the body and that can move around the body. When I was a kid, there was this show where they took a ship and they shrunk it down and they put it in the bloodstream, and it would move around. It would move around through the body, and it was a very interesting show. I may be dating myself in terms of my age here, but I forget what it was called, but it was very exciting. But these types of nanobots and small motors are increasingly being thought of as ways to put things in the body and perhaps do things like target drugs to sites of infection, target cancer cells, other things like that. Any questions about our nanobot for sperm delivery? So I guess what I think is probably the biggest thing, and I sort of have this picture, this slide from when it used to be on the horizon. It's no longer on the horizon. Gene editing is here, and it's having a profound impact on medicine and agriculture. And I think that this is, of all the technologies that I see right now, this one has the greatest possibilities for addressing many of really challenging issues in both human health and in agriculture. So just real quickly to review, back in the days before gene editing, we could cut a gene out, and we could paste that gene into another organism using some fairly crude tools that were not really specific. And what genetic engineering allows us to do is to go in, and rather than transfer a gene from one species to another, we can go in and edit the existing DNA and change the sequence to have the outcome we want. We could eliminate a gene, we could insert a gene, we could modify down to one nucleotide or as many thousands of nucleotides at one time. And these technologies are getting more and more easy to accomplish. So these gene editing tools fall in this family of meganucleases. The one that you probably heard the most about is the CRISPR-Cas9 system. But basically, all of them have the same principle where they have some sequences of nucleic acids. Here, in case of CRISPR-Cas, it's the guide RNA, which will target you to a specific site, kind of like a zip code brings a letter to your mailbox. That guide RNA will drive the CRISPR-Cas complex to the site where you want to make the change. And then when the CRISPR-Cas or the other meganucleases get there, they can make a double-stranded DNA break. So they use a molecular scissors. They make a double-stranded DNA break. And when they do that, they take advantage of the fact that the cell will then try to repair that. And in that repair mechanism, they can do things like they can insert a sequence, they can delete a sequence. And so this is the principle for gene editing. It's very much like if you wrote a paper with your friend, they send the first draft and you can go in and cut and paste and insert anywhere you want in a very specific fashion. But where did these enzymes come from? And in many cases, people already have a poor view of scientists and what they do in the lab late at night. But where did this revolutionary, DNA editing technology come from? Does anyone know? Anyone know? Was this thought up in the lab of an evil scientist late at night? Kind of the scientist you see on the cartoons, or is this something else? Neil.
Well, I remember reading about it, but I know that it's not it's not made up. It's a natural thing, correct?
That's right. You're right, Neil. It is. In fact, the CRISPR-Cas system is a bacterial defense system. And bacteria, if you look at those green spheres on your left, that's Streptococcus pyogenes. They'll give you a nasty infection, and we don't like them at all. But they have their enemies as well. And bacteria are attacked by these other smaller organisms called bacteriophage. And these are examples of bacteriophage actually landing on a bacteria, and they actually insert their genetic material into the bacteria. So the bacteria have developed a system to be able to look for this attack and to be able to chop up any of this foreign DNA. So this is a bacterial defense system that scientists discovered in the natural world. And so if you look, these bacteriophage are pretty interesting little molecules. This is a sort of colorized schematic here, but this is pretty much what they look like. They basically deliver their nucleic acid into a cell. But it kind of reminded me of the War of the Worlds. I don't know if you ever saw the movie, but it kind of reminded me of when the aliens set down on planet Earth and just about wiped us out. But basically, all it is a DNA. It's a device that can target specific areas in the DNA, and you can see in the middle of the green blob here is the guide RNA. That's the zip code that will take the big blue blob, which is a molecular scissors. It'll take it to its location, and if it's a perfect match, it'll cut, and then it can make an edit in that spot. And so this is what that would look like with the guide RNA sitting down. It first unwinds the DNA, and it looks for base pairing. You can see if the base pairing is correct, then you'll see 2 single-stranded cuts will be made there, and then again there. And then at that point, then using the cell's own repair mechanisms, it can insert, delete, make a single change, make multiple changes. And you can edit multiple genes at the same time. They've now shown that they can edit up to 1,000 genes at the same time using this technology. So I have heard a number of people speak about the potential of gene editing, and it's particularly in things like human medicine. So for example, there are many diseases that result from single cell or single gene problems, like for example, sickle cell anemia is a result of a mutation in a single gene. And this young woman is in the last year been treated with cells that have been gene edited, and to repair the error in the gene. And the last I heard, which was not too long ago, she was getting better and better, and the sickle cell disease was being cured. And there are, cancer typically occurs in people because there's a mutation or multiple mutations. And if you can treat adults with this technology, you could potentially have new ways to combat cancer, neurodegenerative diseases, Alzheimer's disease. So the potential is great. So gene editing is here, and it's being used in both agriculture and in human medicine. I'm just going to go through quickly about some of the applications on genetic engineering in animals. We don't have a tree fish yet, but I'm sure that's coming down the pipe sometime soon. But you may have heard of the Aqua Advantage salmon. This is the first genetically engineered animal that's been approved for human consumption in the United States. Anyone know when this fish was developed? It was developed in 1989, and it was put up for approval in the mid 90s, and it took 20 years and multiple studies and reviews before it was finally approved in November 2015. Immediately then, the good senator from Alaska, Senator Murkowski, whose constituents have a huge wild salmon fishing industry up there, thought we should take a little bit more time and think about it. And so for a period of time, it was banned from being sold until they decided how they were going to label it. It's now been approved for sale. It's been sold in Canada for a little over a year now. Just this last week, however, a judge from California has decided we need to look at some more things. So they've decided they have to do another environmental assessment on this particular animal, but it will be able to be sold, and I think you'll see it at limited distribution starting as early as the spring. This particular salmon used the old transgenic approach. It basically has it's just an Atlantic salmon. It has a gene, a growth hormone gene from a Chinook salmon, which makes it grow more uniformly across the year. And that's driven by a promoter from another fish. And so what you have is a salmon that grows 2 to 4 times faster. And so it's much more efficient. And as you may know, we are now overfishing most of our wild fish stocks. And if you look at wild fish harvest, it has been essentially flat for the last 20 years because we have overfished those. And so the increase in consumption of fish is primarily from farmed and cultured fish. And if we're going to continue to eat more fish, we're going to need to find ways to be able to grow those fish in a sustainable way. We certainly cannot do that off the backs of wild populations anymore. That's just the federal judge ruling that came out just this last week. But that's okay. Those things need to be done. They've been done multiple times, but until there's, as long as there's a judge who wants another environmental assessment study, the industry is going to have to do one. You may not have heard of gene edited pigs that are resistant to a major disease in pigs. PRRS is a disease that affects reproduction, and it costs the industry over $100 million a year. And so they've been able to figure out this is a virus, and the virus binds to a very specific molecule. and they know exactly the couple of amino acids on that molecule that the virus binds to. So they've been able to go in and just change those amino acids. The virus can't bind to the cells anymore, can't affect the pig, and it doesn't affect the growth or production of the pig. There's also a pig that's been genetically modified so it expresses phytase in its saliva, and that phytase helps it use phosphorus in its diet more effectively and reduces the phosphorus in its manure by about 50%. And if you live in the Chesapeake Bay watershed like we do, phosphorus, nitrogen, phosphorus, and sediment are the three main things. And so a technology that could reduce phosphorus in the waste seems like a win-win. Here's a group of scientists, the same group that cloned Dolly the sheep back in the 90s, developed a chicken that resists transmission of bird flu and slows down the spread, allowing farms to get a handle on the disease and isolate it and depopulate those flocks before it can be spread. USDA scientists developed a cow they named Annie that's resistant to mastitis. And if you know the dairy industry, mastitis is a common infection that occurs, common disease that occurs on dairies. And this animal is resistant to mastitis. From a welfare standpoint, if in the Holstein breed and many other breeds where the animals have horns, we do a procedure called dehorning when the animals are very young. It's a fairly quick and mostly painless procedure, but people perceive it as being an animal welfare issue. And so what if you could genetically remove the horns from an animal or breed that has horns? That would certainly be a win for everybody. We know the sequence of the gene that gives you horns or scurs or polled. That's known. It's been known for a long time. And so scientists at University of California went in and just edited the horn gene. They made a couple of changes. They deleted 10 nucleic acids, 10 nucleic acids, and they added 200. And they basically turned a horned Holstein bull into a polled Holstein bull. And these are two of his calves, and they're polled, and all their offspring will be polled. And so none of their offspring will have to go through that dehorning procedure. They've also, we know a lot about regulation of growth and some of the genes that regulate growth. And this particular animal was engineered to grow more muscle. And you can see the carcass difference between these two animals by making a change in one particular gene. They were able to get a carcass with much more meat per carcass. If you get more meat per carcass, fewer animals to raise the same amount of meat, fewer animals results in less feed, less space, less manure, and less environmental impact. So whether these technologies are going to be adopted or not is still up in the air. Certainly, there's plenty of scientists can come out and support gene editing and genetic engineering and talk about the benefit. But until we change hearts and minds of the consumer, that I think that we're going to be slowed. But I'm an optimist. I actually think that as I interact with students here at Penn State and talk to various groups, that young people care more about the environment than they fear technology. Many of these students in high school were doing DNA technologies, and so they come they come with a different set of goals and aspirations, and technology doesn't scare them. So what is the farm of the future going to be? Well, it could be this floating farm in the Netherlands that's built on pontoons and won't be affected in case of a flood. It'll just rise with the water, and I'm not sure how widely this will be adopted, but there are a number of these around the world. Certainly, we could see some changes in housing. These 7-story high rises in Guangxi province in China these are pig farms with 1,000 pigs per floor, 7 stories. These are 1, 2, 3, 4 times 7. That's a lot of pigs on that hillside right there. So there'll be some changes in facilities, but I think the biggest changes that are going to occur in terms of how we monitor our animals and the data we collect on our animals. And so in the last couple slides, I'm just going to show you some slides I grabbed off the internet that reflect some of the work that we're trying to do here, where we're going to be deploying technology. And in that case, and in this case, I'm talking about things like sensors, both sensors that can be, for example, worn around the neck or implanted in the ear or actually implanted in the body, or sensors that will be able to be put in the milking line. including things like cameras that will be able to monitor animals and recognize animals based on their features. And these sorts of technologies are going to be linked using wireless technology. They're going to incorporate artificial intelligence, which is going to learn, be able to learn what normal physiology is, so they can then detect deviations from normal physiology, which occurs in the onset of disease. These algorithms are then going to be able to work through the internet to either alert farmers or veterinarians that animals need attention, or even things like change that animal's location or change the feed that's provided to that animal. Inline sensors in milk are going to be able to, are currently able to monitor a lot of about the physiology of the animal beyond just milk production and protein and fat, but things like their metabolic status and whether they're pregnant or not. This is the kind of technology that I see being developed rapidly, and we're going to see more and more of this. There's going to be a lot more about treating the cow as an individual, including things like caring about how they're interacting socially with each other, and also how they're interacting with humans, and how they're interacting with their environment, to try to understand what is the optimal cow day. And we know a lot about how to make a cow happy, and we like to be able to do that on every cow. And in order to do that, you have to have the ability to collect big data and make real-time decisions. And so we're going to see a lot more technology that's designed to be monitoring these animals in real time. I also mentioned that milk is a tremendous tissue to be able to sample. We sample milk 2 to 3 times a day. And as we look into that milk, we can learn a lot about the state of the animal's physiology and production. And so a number of the companies that produce milking systems are developing inline sensors for everything from pregnancy checking to somatic cell count to progesterone, to ketones and other things that give indication of the physiological status of that animal. So with that, I know it's been a whirlwind tour here on the animal biotechnologies, and I've only scratched the surface here, but I would be happy, and I have a few minutes. John, my next meeting starts at 5. I do have to get from here to home by then, but that's only a 10-minute drive. So I have a few minutes if people have some questions they'd like to ask, or if they have some things that they're hearing about that they want to talk about.
Absolutely. Very, very, very awesome stuff, Dr. Ott, and we appreciate that. Does anybody have any questions about anything that Dr. Ott covered or that he didn't cover that you know of that you're curious about? As you said, we have a couple minutes for those. Feel free to unmute yourself if you're able, or if not, you can use the Q&A pod or pose them in the chat and we can read them out loud.
So Ryan says he doesn't have a mic. So Ryan, go ahead and type then. Anything I said that you thought was different than you heard or raised some concern?
Dr. Ott, this is from Ryan. Do you think that the test trial periods for genetically modified organisms used in food production are actually needed to ensure safety, or mainly to appease public concern?
I think they're certainly important to ensure safety. And if you think about it, the first genetically modified foods were approved in 1995, 96, and we've been consuming them for some time. In fact, if you think about pigs and poultry, they've been eating essentially nothing but genetically engineered corn and soy for the last almost 30 years now, 25 years. And so we've been doing feeding trials on an epic level. And certainly those products have been in our own foods that we consume. I think that we have to, when people say I'm against GMOs or genetic engineering, you can't be against a technique. You can be against an application of a technique. Let's say I decided to put cyanide in an apple or in a something and make it toxic. That you could be against that, but you can't be against the technique. So what you try to what I try to do is try to focus people on what is it that you're concerned about? Okay, is it safety? All right, so let's look at the data on safety. These technologies can be used to actually improve health, improve the quality of the product, improve animal welfare. So don't throw the technology out, but certainly challenge the individual application. And I think that's important. And I think that on the USDA side, that we've gotten the path to approving these fairly well down. But on the animal side, it's been a bit more rocky of a road. People tend to be much more concerned about gene edited animals than they are about gene edited plants. So I think the safety is essential. That has to be job one. But I think what we learned in the first round in the 90s is you have to actually inform the public as you go along. You can't do the old ta-da, here it is. You actually have to prepare the ground, let them know what's being done, let them know how it's being tested, share that in a transparent way. Talk about the benefits. What are the benefits of using these technologies? How about if we reduce the pressure on the Atlantic salmon population because we could grow them inland in inland tanks and recirculating tanks and not have to rely so much on wild caught? We don't talk enough about the benefits. We always imagine the worst thing that could happen, and we spend all our time talking about that and not enough time talking about the real benefits that these technologies, not just promise, but have already delivered. So for example, BT varieties of plants have tremendously reduced the spraying of insecticides. It's been a big win. But, someone says, well, the insects will develop resistance. Yes, that always happens. That's the way life works. Whenever you have one solution, the evolution always allows change. And so you have to develop a slightly modified version of BT. But they've been deploying the BT eggplant in Southeast Asia. And the pesticide poisonings have greatly dropped because little kids were spraying these eggplants every day, unprotected, with these toxic pesticides that aren't allowed to be used in this country. Farmers are making a really good income now. And so now they're being able to put money aside and send their kids to school. These things are having real positive benefits. Glyphosate resistant crops have aided the adoption of no-till practices that have reduced erosion in areas that were susceptible to erosion. So there's plenty of good stories. We have to own up and be really transparent with what we're doing, and we can't just spring these on the public. We have to bring the public along. The public has to agree that this is a good thing, and we have to bring them along with us.
We had a question from Holly in the chat. The sensing technologies that you covered, do you see those being widely available and utilized in all agricultural livestock, from poultry to swine to bovine, et cetera?
Yeah, I do. And I talk a lot about dairy because I work with dairy cows mostly. And of course, dairy are an animal that we handle a lot every day, and they're more amenable to some of these things. But just recently, Penn State had a big news story about bioprinting sensors that you could stick on the skin that could give you readings of certain hormones being produced in the individual. Whenever you're working with food producing animals, implantables are a challenge just because you got to recover them at some point. And so I'm really excited about what we can do now with cameras and remote sort of technologies that don't involve you putting anything in the animal, but provide you information about the physiological status of that animal. So I think they're going to be applicable. Certainly on poultry, it would be barn scale type stuff. You don't want to, put anything on an individual chicken. That's just too much work. But you want to be able to monitor the animals, their health, their well-being, their feed intake, those sorts of things. So I'd say in just about every type of animal production system. I'm looking.
More question from Ryan regarding social media. Do you see that as a help or a hurt as far as informing public and consumers about the benefits of genetically modified organisms, or more so in spreading the bad science?
Yes. It is both a help and a hurt. And so you'll see, I posted there my Twitter handle. That's the only social media other than LinkedIn that I'm on. And I use it as a way to to funnel information that I need to see to me. So I follow thought leaders in various areas, including people who disagree with these technologies, because I always want to know what those arguments are. I always want to understand why people are against something that I think is something that's a positive. I find most of my cutting edge science now via Twitter, and I think Twitter is good because you can follow the people that you want to follow. I don't like Facebook at all because Facebook tries to figure you out and show you things that you already like, and so that's a great way to never change, is have a platform that tries to just keep showing you things that you agree with. So I like to be challenged. I like to hang out with people that disagree with me, and I like to follow people that disagree with me, because I know in the end my thinking is going to be better, and I won't be blindsided by something that I just didn't have the intelligence to figure out. So they are a great good, and they are also a problem. And we're in the first generation of these, and so we'll figure it out over time. But I think there are real challenges especially if you try to go out there and change hearts and minds on social media. So what you'll see with my Twitter feed, there's nothing personal there. It's just about providing information. So I try to find good information on things that need more good information, and I put that information out there, and I don't engage in any debates or arguments on Twitter. I just try to bring light and not heat to the issue.
Very good advice to end on, Dr. Ott. Thank you so much for a great presentation. Teachers, you had some great questions. Thank you for asking them. I think Dr. Ott has given you more than enough fodder to get some conversations started in your classrooms with students, and maybe some new ideas about some emerging technologies to cover with students that you may not have been aware of already. So with that, I think we better let Dr. Ott head for home, as well as let the rest of you get on with your much deserved evenings. So thank you on behalf of all of us, Dr. Ott. We appreciate you sharing your expertise and insight. Teachers and attendees, we greatly appreciate you giving an hour of your valuable time. So with that, you may begin to adjourn and log off. I'll remain on till everybody else is gone.
Thanks, guys. Take care.